10 volcanic islands rise from the Atlantic Ocean, hundreds of kilometers west of the African mainland. Today, they form Cabo Verde. But within the DNA of Cabo Verdean people lies evidence of one of the Atlantic world's most remarkable population histories. The story connects Western Africa with Portugal and the wider Iberian Peninsula. It reflects migration, colonial expansion, forced movement, family formation, adaptation, and centuries of travel between islands. Yet Cabo Verdean DNA cannot be reduced to one percentage. Different islands preserve different genetic histories. Maternal lineages tell a strikingly different story from paternal lineages. Modern genomes reveal strong connections to Senegambia and Iberia while also recording what happened after those populations met. So where do the people of Cabo Verde come from? What can DNA genuinely tell us about their ancestry? And where do the limits of genetics begin? To answer those questions, we have to start before there were Cabo Verdeans. The genetic history of Cabo Verde begins with geography. The archipelago lies in the Atlantic west of Senegal, separated from the African mainland by hundreds of kilometers of open water. Cabo Verde contains 10 principal islands, 9 of them inhabited today, and its population history differs from that of most places in Africa or Europe. Present archaeological and historical evidence does not establish a permanent population living on the islands before Portuguese Atlantic expansion. Modern Cabo Verdean society therefore developed comparatively recently, during an era for which significant written documentation also survives. Portuguese navigators reached the islands during the 15th century, and permanent settlement began on Santiago around 1462. That date matters. It places the emergence of Cabo Verdean society at the beginning of sustained European expansion into the Atlantic. Santiago became the first major center of settlement. Ribeira Grande, now known as Cidade Velha, grew along the island's southern coast. Its location connected Cabo Verde with Portugal, the Western African coastline, and eventually an expanding network of Atlantic routes. But the populations participating in this new society did not arrive under equal circumstances. European newcomers were predominantly Portuguese, and many of the earliest settlers were men occupying positions of political, commercial, landholding, or religious authority. Africans arrived through several kinds of movement. Some came through commerce, employment, regional travel, or later voluntary migration. Many others arrived through colonial systems of forced migration and labor connecting the islands with the nearby African coast. Senegambia, the region encompassing much of present-day Senegal and the Gambia, was particularly important. Connections extended southward toward what is now Guinea-Bissau and other parts of the upper Guinea coast. The African population involved in this history was never a single uniform community. People arrived from societies speaking different languages and carrying different political, cultural, and family traditions. Some came from areas associated with Mandinka-speaking communities. Others were connected with Wolof-speaking populations. Still others came from different regions of Western Africa. Modern genetics can identify affinities with some present-day populations, but it cannot reliably assign every historical ancestor to an exact ethnic identity. That distinction is fundamental. A present-day population used as a genetic reference is not identical to the communities that existed 5 centuries ago. Borders changed. Populations migrated. Languages evolved. Political identities appeared, disappeared, or transformed. Genetic studies therefore estimate relationships and affinities. They do not provide perfect historical labels. Santiago became Cabo Verde's earliest major demographic center, but the rest of the archipelago did not develop simultaneously. Fogo was settled relatively early and maintained strong historical links with Santiago. Other islands developed substantial permanent populations later and under very different economic circumstances. Santo Antão and São Nicolau follow their own demographic paths. Boa Vista and Maio were shaped by different combinations of livestock raising, agriculture, maritime activity, salt production, and migration. São Vicente became a major population center much later, especially with the growth of Mindelo as an Atlantic maritime port. São's modern expansion belongs largely to a still later period. Brava developed from a comparatively small founder population and remained closely connected historically with Fogo. These differences eventually became visible in DNA. An island is not an absolute biological barrier. People have always crossed the sea between Cabo Verde's islands. But before modern transportation, stretches of ocean could reduce how frequently individuals from different communities met and formed families. If one island received proportionally more newcomers from Europe, while another received more African-descended migrants, their average ancestry could gradually diverge. And when an island began with a relatively small founder population, chance could make certain lineages unusually common over time. Severe droughts, food shortages, disease outbreaks, emigration, and economic changes could alter population size again. Each of these demographic events had the potential to leave a genetic trace. That is one reason Cabo Verde has become so interesting to population geneticists. Its population formed recently enough for researchers to compare DNA with colonial documentation, settlement histories, migration patterns, linguistics, geography, and genealogy. Written records tell us about institutions, ports, ships, settlements, property, and colonial administration. DNA answers a different question. It reveals which ancestral populations actually contributed descendants to later Cabo Verdean generations. Those two records do not necessarily produce identical pictures. Thousands of people might move temporarily through an Atlantic port without leaving a substantial genetic legacy there. A much smaller community that remained and formed families for generations could contribute disproportionately to the modern population. That is why Cabo Verdean population history cannot be reconstructed from shipping records alone. But neither can it be reconstructed from genetics alone. The clearest picture emerges when the two types of evidence are compared. The islands began without the population we now recognize as Cabo Verdean. Then people from Africa and Europe encountered one another within the unequal structures of Portuguese colonial expansion. Over generations, their descendants created families, communities, local traditions, and eventually a population with a history of its own. The ancestral sources were elsewhere. The Cabo Verdean people were formed in the Atlantic. When people hear the phrase DNA ancestry, they often imagine that scientists can divide a person into exact biological percentages. Human genetics is far more complicated. Nearly all human DNA is shared across our species. Population geneticists instead examine large numbers of genetic variants that occur at somewhat different frequencies in different populations. When researchers describe part of a Cabo Verdean genome as having greater affinity with Western Africa or Iberia, they are not identifying an African gene or a Portuguese gene. No such single markers exist. Researchers compare patterns across hundreds of thousands, or sometimes millions, of positions in the genome with patterns found in reference populations. Some stretches of DNA may resemble variants more commonly observed in Western African reference samples. Other stretches may more closely resemble people sampled in Portugal, Spain, or surrounding parts of southwestern Europe. Statistical models then estimate the ancestry proportions most consistent with those patterns. This is the foundation of genetic admixture analysis. Cabo Verde is particularly informative for this research because some of its major ancestral sources came from geographically separated populations and because large-scale population formation began only several centuries ago. One landmark analysis, published in 2012, studied 845 people from 6 Cabo Verdean islands. Researchers examined autosomal markers, X chromosome markers, and markers on the Y chromosome. Autosomal DNA comes from both parents and captures information from many branches of a person's family tree. Across the full sample, the researchers estimated an average West African autosomal ancestry of about 57%. Most of the remainder was associated with European ancestry, with Iberia representing the strongest relevant European source, but the figure of 57% should never be treated as the universal ancestry of a Cabo Verdean person. It was an average from a particular scientific sample. Island averages differed. In that study, Santiago had the highest mean West African autosomal ancestry at about 65%. Boa Vista was about 59%. Santo Antão, São Vicente, and São Nicolau averaged close to 56%. Fogo was approximately 53%. Even these figures are not permanent genetic labels attached to the islands. Results depend partly on sampling, genetic markers, reference populations, and statistical methods. Individuals living on the same island can also differ substantially. A person from Fogo, for example, can have more West African ancestry than a person from Santiago, even though the average values of the two sampled island populations moved in the opposite direction. Population averages describe statistical patterns. They do not define individuals. More recent genome-wide research has reinforced the broad picture while giving scientists greater geographic resolution. A 2023 study examined genetic and linguistic information from participants connected with all 9 inhabited Cabo Verdean islands. The strongest ancestral affinities again pointed towards southwestern Europe and Western Africa. On the European side, Iberian reference populations were dominant. On the African side, particularly strong connections appeared with Senegambian populations, especially Mandinka and Wolof reference groups. This is significant because the result corresponds closely with historical evidence concerning Cabo Verde's early connections with the Upper Guinea region. Portuguese Atlantic expansion linked the islands extensively with areas near modern Senegal, the Gambia, and Guinea-Bissau. Genetic analysis independently identifies the same broad region as especially important to the ancestry of modern Cabo Verdeans. Yet historical movement and genetic contribution do not always correspond neatly. Records document people from additional Western and Central African regions moving through Cabo Verdean Atlantic networks. Modern genomes, however, do not preserve equally strong contributions from every documented population. One likely reason is that passage through Cabo Verde was not the same as permanent settlement. Many people continued onward to other destinations. Some spent only limited periods on the islands. Others did not establish family lines that remained represented in later generations. Genetics therefore records reproductive contribution rather than total population traffic. The genome also changes in appearance across generations because of recombination. When two previously separated populations begin forming families, the DNA inherited from each ancestry source initially appears in relatively long chromosome segments. With every new generation, chromosomes are reshuffled. Those ancestral segments gradually become shorter. After many generations, a person's chromosomes can contain hundreds of segments originating through different ancestral pathways. By examining the size and distribution of these segments, scientists can sometimes estimate approximately when population mixture occurred and whether later migrations introduced additional ancestry. Cabo Verdean genomes therefore contain information not only about geographic origins, but also about population history through time. The most important conclusion is that there is no scientifically meaningful single genetic formula for Cabo Verdeans. Instead, the evidence describes a population formed primarily through substantial Western African and Iberian ancestry, followed by centuries of island-specific migration, marriage patterns, founder effects, demographic change, and recombination. The genome is not a simple percentage chart. It is a mosaic assembled by history. One of the clearest genetic windows into Cabo Verde's past comes from mitochondrial DNA. Most of our DNA exists inside the cell nucleus. Mitochondria, however, contain a very small genome of their own. Mitochondrial DNA is normally transmitted from mother to child. Both sons and daughters receive it, but daughters are generally the ones who continue transmitting that same mitochondrial lineage to later generations. Researchers can therefore use mitochondrial DNA to follow one narrow path through a family tree. Your mitochondrial DNA came from your mother. She received it from her mother. She received it from hers. And the chain continues backward. This does not represent all of a person's female ancestors. It represents only one direct maternal line. But when scientists examine many people from the same population, mitochondrial lineages can reveal important demographic patterns. A major study published in 2002 analyzed mitochondrial DNA from 292 people sampled across 7 inhabited Cabo Verdean islands. Its conclusion was striking. The maternal gene pool was overwhelmingly associated with Western African lineages, with almost no detectable Portuguese mitochondrial contribution in the combined sample. This does not mean European women were entirely absent from Cabo Verdean history, or that none left descendants. It means that among the direct maternal lines preserved in the population studied, African ancestry overwhelmingly dominated. Historical demography offers a compelling explanation. Early Portuguese migration to Cabo Verde was strongly male-biased. European women were far less numerous than European men during key phases of early settlement. African women, meanwhile, became central members of the emerging island communities. Over generations, their direct maternal lineages became deeply represented in the population. Imagine an African woman arriving on Santiago during the 16th century. Written documents may preserve little about her. Her original name might be missing. Her birthplace might have been recorded only vaguely. Her language, family history, and community identity might never have entered an archive. But if she had a daughter, and that daughter had a daughter, and the maternal line continued uninterrupted, descendants centuries later could still carry mitochondrial DNA inherited through that chain. Genetics cannot tell us what she thought. It cannot recreate her voice. It cannot tell us the circumstances of her relationships or the details of her daily life. But population genetics can demonstrate that women like her were fundamental biological founders of later Cabo Verdean generations. That matters because colonial archives preserve people very unevenly. Governors, merchants, clergy, landowners, ship captains, and administrators appear regularly in surviving documents. African women living under dependent or unequal legal conditions may appear only in church registers, property records, household documents, or fragmentary references. Some may not appear individually at all. DNA cannot replace missing historical testimony, but it can reveal demographic significance that written records sometimes obscure. The Western African character of many maternal lineages also connects Cabo Verde strongly with Senegambia and the Upper Guinea coast. Earlier mitochondrial studies identified similarities with Western African populations, while later genome-wide work found particularly strong relationships with Mandinka and Wolof reference populations. Again, terminology requires care. A particular mitochondrial lineage should not automatically be labeled Mandinka DNA or Wolof DNA. Many lineages are much older than present-day ethnic identities and may occur across neighboring populations. Reference groups are useful statistical comparisons, not exact reconstructions of 15th-century societies. The strongest conclusion is therefore geographic rather than narrowly ethnic. Cabo Verde's direct maternal ancestry is deeply rooted in Western Africa, with Senegambia playing an especially important role. Mitochondrial DNA also demonstrates why no single genetic marker represents a person's total ancestry. Someone can have hundreds of female ancestors across the generations while carrying mitochondrial DNA from only one uninterrupted maternal chain. Your father's mother contributed autosomal DNA to your father and potentially to you, but her mitochondrial genome normally stops at your father. Your mother's father's mother also contributed ancestry to your family tree, yet that mitochondrial lineage does not reach you through the male link. Mitochondrial DNA, therefore, does not summarize all female ancestry. Its value comes from precisely the opposite property, its unusually narrow inheritance path, and that makes Cabo Verde's pattern particularly informative. If direct maternal lineages are overwhelmingly Western African, while the overall autosomal genome contains a substantial European component, then women and men from the major source populations clearly contributed differently during population formation. The maternal record is therefore central to the story. It tells us that African women played an extraordinary demographic role in founding Cabo Verde's later generations. Their descendants became part of communities that eventually developed a distinct Cabo Verdean identity. DNA preserves one small biological thread connecting those women with the present. It cannot tell their complete story, but it provides powerful evidence that their descendants endured. If mitochondrial DNA traces one direct maternal line, the Y chromosome can reveal one direct paternal line. Most males inherit a Y chromosome from their father. That father inherited his from his father, creating a line that can extend backward through many generations. Like mitochondrial DNA, the Y chromosome represents only one branch of a much larger family tree. Yet at the population level, Y chromosome patterns can reveal remarkable historical differences. In Cabo Verde, they contrast sharply with the mitochondrial record. A 2003 analysis found substantial representation of paternal lineages associated with Europe and the broader Mediterranean, alongside paternal lineages associated with Western Africa. The difference became especially clear in the larger 2012 population study. Using one four-way ancestry model, researchers estimated that roughly 68% of the sampled male line contribution was associated with Iberian reference populations and approximately 27% with Western Africa. The model assigned much smaller percentages to North African and Sephardic Jewish reference populations. These numbers should be interpreted carefully. They are statistical population estimates produced by a particular model. They do not mean that every Cabo Verdean man has the same paternal ancestry, and the proportions varied greatly between islands. Still, the contrast with mitochondrial DNA is unmistakable. Direct maternal ancestry was overwhelmingly Western African. Direct paternal ancestry showed a much stronger European contribution. This is known as sex-biased admixture. The idea is straightforward. Men and women from the ancestral source populations did not contribute equally to the generations that followed. European men became direct paternal ancestors at a substantially higher rate than European women became direct maternal ancestors. African women, meanwhile, became direct maternal ancestors at a considerably higher rate than African men became direct paternal ancestors. Historical records help explain the pattern. European settlement during important early periods was overwhelmingly male. At the same time, large numbers of African women were incorporated into island society through colonial systems characterized by very unequal legal and social conditions. Population formation consequently occurred within a society where ancestry, gender, status, and political power were deeply intertwined. The X chromosome provides another independent source of evidence. Women typically inherit 2 X chromosomes, while men normally inherit 1 X chromosome from their mother. Because the X chromosome spends proportionally more generations being transmitted through women than through men, sex-biased ancestry can produce different genetic patterns on the X chromosome compared with the autosomes. That is exactly what scientists observe in Cabo Verde. A 2022 Genome-Wide study analyzed 563 individuals from 4 regions of the archipelago. Average African ancestry in that data set was about 60% across the autosomes but approximately 76% on the X chromosome. That difference strongly supports a greater female contribution from Western African populations and a greater male contribution from European populations. When mitochondrial DNA, Y chromosome lineages, X chromosome ancestry, and autosomal DNA all point in compatible directions, confidence in the demographic interpretation becomes much stronger. But genetics has limits. It would be misleading to reduce this history simply to a statement that European men and African women form families. The society in which those families emerged was deeply unequal. Some relationships were marriages or enduring partnerships. Others developed within circumstances of legal dependency and major imbalances of power. DNA cannot distinguish between those situations for an individual conception. A chromosome can record biological inheritance. It cannot reconstruct the social circumstances surrounding a relationship. Historians therefore need church registers, court documents, property records, family records, colonial legislation, and other evidence to understand the human context behind the demographic pattern. Genetics establishes the imbalance. History helps explain the society that produced it. The Y chromosome also demonstrates why a single lineage should never be confused with a person's complete ancestry. A Cabo Verdean man might carry a paternal lineage associated statistically with Iberia, while most of his autosomal genome is West African related. Another could carry a West African associated Y lineage while having substantial Iberian autosomal ancestry. The Y chromosome represents only one father-to-father path. The same limitation applies to mitochondrial DNA. These markers are valuable precisely because their inheritance is so specific. Together, the evidence tells a remarkably consistent story. Cabo Verde's overall ancestry is substantially Western African and European. Its direct maternal lines are overwhelmingly Western African in the major mitochondrial study. Its paternal lines contain a much larger European contribution, and its X chromosome independently shows stronger African ancestry than the autosomes. This is more than information about geographic origin. It is biological evidence of how a colonial-era society reproduced itself across generations. Santiago and Fogo demonstrate why Cabo Verdean ancestry cannot be summarized with one national percentage. The islands lie relatively close to one another. Both were populated early. Fogo's settlement was historically connected with Santiago, yet their modern genetic patterns differ substantially. In the 2012 autosomal study, Santiago had the highest mean West African ancestry among the 6 islands sampled, approximately 65%. Fogo had the lowest, around 53%. Again, those are population averages rather than boundaries between individuals. The Y chromosome reveals an even sharper difference. The same study's paternal ancestry model estimated considerably more West African paternal contribution on Santiago, while Fogo showed a much larger Iberian-associated component. How could 2 nearby islands with interconnected histories become genetically distinct within only a few centuries? The answer is demography. Santiago became Cabo Verde's earliest major administrative, commercial, and population center. Ribeira Grande connected the island repeatedly with Portugal and Western Africa. New migrants arrived. People moved onward. Some remained permanently. Communities expanded and changed. Santiago therefore experienced repeated episodes of population movement and mixture. Fogo followed another path. Although some of its early settlers came from Santiago, later migration, landholding patterns, economic structures, local marriage networks, and population size developed differently. Even relatively modest demographic differences can become genetically important when a population is small. Suppose an island contains only several thousand people and a few dozen new families arrive. If those families have many descendants, their genetic contribution can become substantial within several generations. Founder effects amplify this process. An island may begin with hundreds rather than millions of settlers. Some families happen to become very large. Others become less represented. The genetic composition of the descendants can therefore differ from the ancestry balance of the original population. Recent genome-wide research adds further detail. The 2023 analysis suggests that Cabo Verdean admixture was not one single event occurring at one moment. Instead, individual islands experienced different sequences of population formation and later migration. On Santiago, researchers found evidence compatible with early mixture between Senegambian-related and southwestern European populations, together with later contributions. Fogo appears to preserve another trajectory, beginning partly through movement from Santiago but developing its own demographic structure afterward. The distinction between port traffic and genetic contribution is especially important. Historical records may document large numbers of people moving through an island. That does not mean all of them contributed equally to future generations. Genetic ancestry depends on who stayed, formed families, and left descendants. Movement and ancestry are related, but they measure different things. Geography also reinforced island differentiation. Today, a voyage between Santiago and Fogo may seem routine. Centuries ago, travel depended on sailing conditions, vessels, economic resources, and maritime networks. Movement certainly occurred, but it was not sufficient to make the islands one completely mixed population. Families frequently formed within local social and geographic networks. Over generations, those patterns accumulated. This is why island-level averages need to be interpreted demographically rather than racially. Santiago does not possess an African essence. Fogo does not possess a European essence. Both populations contain substantial ancestry from Western Africa and Europe. What differs statistically is the average balance and the history through which that balance developed. The distinction tells us nothing about cultural authenticity. A Cabo Verdean family from Fogo with relatively greater Iberian ancestry is not automatically more Portuguese culturally. A family from Santiago with relatively greater West African ancestry is not automatically more African culturally. Cabo Verdean culture developed through language, place, family, religion, migration, music, food, politics, and shared historical experience. Genetics measures biological ancestry. It does not measure cultural membership. Santiago and Fogo demonstrate how quickly detectable population structure can form when geography and social history operate together. 5 centuries is extremely recent in evolutionary terms. Yet within that period, neighboring islands acquired measurable genetic differences. That is one reason Cabo Verde has become so valuable for studying recent human admixture. Scientists can investigate demographic processes that happened relatively recently and compare their models with unusually rich historical evidence. Santiago and Fogo provide different answers. Together, they make one conclusion unavoidable. There is no single Cabo Verdean ancestry profile. To understand the DNA, you have to understand the island. Cabo Verde is one country. Genetically, however, its 9 inhabited islands do not form one perfectly uniform population. Genome-wide analysis of people connected with the entire inhabited archipelago has revealed shared Cabo Verdean ancestry alongside clear island-level structure. This is exactly what population genetics predicts when communities remain partly separated for many generations. One relevant concept is isolation by distance. People living closer together generally have more opportunities to meet and form families than people separated by greater distances. On a continent, the effect may unfold gradually. Across an archipelago, stretches of ocean can make the boundaries more noticeable. Before modern ferries, aircraft, telecommunications, and reliable maritime transport, even relatively short journeys at sea required time, resources, and favorable conditions. People moved between islands, but most potential partners were still likely to come from communities relatively nearby. Over generations, this could produce genetic differentiation. Cabo Verde adds another important factor. The islands were not permanently populated at exactly the same time. Santiago became the earliest major center. Fogo developed soon afterward. Several other islands expanded significantly later. São Vicente's major demographic rise was closely connected with Mindalo's development as a maritime center. São's substantial modern population growth also occurred relatively late. Brava, Maio, Boa Vista, São Nicolau, Santo Antão, and the remaining islands each followed distinctive combinations of settlement, emigration, isolation, and economic change. Every island, therefore, began with a somewhat different founder population and subsequently received different numbers and types of newcomers. Founder effects can make these differences surprisingly important. Imagine a new settlement created by 50 families. One uncommon genetic lineage happens to be carried by several families that later become very large. Within only a few generations, the lineage could become far more common than it was in the source population. No biological advantage is required. Chance alone can produce the result. Population contraction can intensify the same process. Cabo Verde experienced repeated periods of severe drought, food shortage, and major emigration. Whenever a population becomes smaller, some family lines become less represented while others account for a greater proportion of the next generation. Geneticists refer to particularly strong reductions as population bottlenecks. Founder effects, migration, geography, and demographic contraction can therefore produce detectable island structure within surprisingly short periods. The 2023 Genome-Wide study revealed particularly strong African affinities with Senegambian reference populations. Mandinka-related ancestry was highly important across the archipelago. For Brava, Fogo, Santiago, Maio, and Boa Vista, Mandinka reference samples accounted for nearly all the African haplotypic affinity detected under the researchers' principal models. On São Nicolau, São Vicente, and Santo Antão, Mandinka-related ancestry remained important, while Wolof-related ancestry appeared more clearly as well. This does not mean those islands can be assigned simple ethnic labels. Modern Mandinka and Wolof samples are being used as proxies for broader historical relationships. The Western African population landscape has itself changed across 5 centuries, so the strongest conclusion remains regional. Senegambian populations made a particularly important contribution to Cabo Verdean ancestry, and the relative patterns differ between islands. European ancestry varies too. Iberian affinity dominates the major European signal, which agrees strongly with Portuguese settlement history. Yet subsequent migration and island-specific founder events change proportions locally. Perhaps the most remarkable feature is how much structure developed across such small geographic distances. 2 Cabo Verdean islands can be visible from one another under favorable conditions. Yet generations of local family formation still produced measurable differences. That does not mean each island became a separate biological population. Genetic boundaries remained porous. A better image is a network. Some islands exchanged people frequently. Others were less connected. Particular historical periods encouraged movement. Others restricted it. The genome records those shifting routes and barriers. Similar island-level patterns can appear in surnames, dialects, migration histories, settlement traditions, and family networks. That is why genetics becomes especially powerful when interpreted alongside social history. DNA can show that communities differed. History helps explain how they became different. A national average hides much of the structure. Island-level analysis brings it back into view. What emerges is not 9 unrelated peoples. It is 9 connected population histories woven into a broader Cabo Verdean one. One of the most fascinating developments in recent Cabo Verde research has been the comparison of genetic diversity with linguistic diversity. At first glance, DNA and language have very little in common. Genes are inherited biologically. Language is learned socially. No child inherits Kriolu vocabulary through a chromosome. Anyone can learn a language regardless of ancestry. Yet genes and language both move through communities. Because of that, they can sometimes preserve parallel traces of the same population history. Cabo Verdean Criolu developed through centuries of interaction between Portuguese and African linguistic traditions. It is not broken Portuguese, nor is it simply an African language covered with Portuguese vocabulary. It is a fully developed linguistic system that emerged from the unique social history of the islands. Criolu varieties differ from island to island. Pronunciation changes. Vocabulary differs. Grammar, rhythm, and usage can vary. Those differences reflect settlement, communication networks, island connections, and the histories of individual communities. Genetic variation followed some of those same geographic pathways. For much of Cabo Verdean history, people generally learned language from parents, relatives, neighbors, and local communities. Those were also the social networks in which many families formed. An island with less migration could therefore become somewhat distinctive linguistically, while also becoming somewhat distinctive genetically. The genes did not cause the language. Language did not cause the genes. Geography and community structure influenced both. The 2023 Life Study found a meaningful relationship between genetic and linguistic differentiation across Cabo Verde. Participants' genetic and linguistic similarities partly reflected the geographic distribution of their islands of birth, a pattern the researchers interpreted in relation to isolation by distance. The relationship was not perfect, and it should not have been. Language can spread without large-scale migration. A pronunciation can become fashionable. A word can cross an island in years. A prestigious speech form can be adopted by people who share no recent biological relationship with the community where it began. Genes cannot spread that way. They require biological reproduction. Language is therefore much more flexible than genetic ancestry. But both can still preserve historical structure. Criolu also demonstrates why identity cannot be reduced to ancestry percentages. Imagine 2 Cabo Verdeans raised in the same community. One happens to have somewhat more Iberian-related ancestry. The other has somewhat more West African-related ancestry. They may speak the same Criolu variety. They may share food traditions, music, humor, religion, neighborhood memories, and family customs. Their genomes differ statistically. Their culture may be profoundly shared. That is one reason genetics should never be confused with simplistic ideas of race. African, and European are enormous geographic categories. Africa contains extraordinary genetic diversity. Senegambian populations are not interchangeable with every other African population. Europe contains substantial regional structure as well. For Cabo Verde, terms such as Senegambian related and Iberian related are more informative than broad continental labels. Even those remain approximations. The same principle applies to language. Saying Kriolu contains African influences tells us less than identifying particular connections with Mande languages, Wolof, Temne, or other regional linguistic traditions. The closer the analysis gets to historical evidence, the less useful simple continental boxes become, and Cabo Verdean culture ultimately became more than a collection of source influences. Kriolu is one example. Morna is another. So are Caladira, Batuk, Funana, local food traditions, maritime practices, religious customs, migration traditions, and memories shared across generations. No genetic program can assign percentages to those things. Culture is not inherited according to Mendelian rules. A cultural practice can survive even if the descendants of its earliest practitioners become numerically small. A language can become dominant even if its original speakers were a minority. Music can combine influences in ways that no ancestry algorithm could calculate. This is where Cabo Verde's story moves beyond admixture. At first, people came from different regions. Over generations, their descendants developed communities rooted in the islands themselves. Criolu became one of the clearest expressions of that transformation. The Atlantic encounter influenced both biological and linguistic history. But language eventually became independent of individual ancestry percentages. One person could inherit predominantly African-related ancestry and speak Kriolu. Another could inherit more European-related ancestry and speak exactly the same language. Culture crossed the genetic differences that history produced. Genes can reveal migration and biological connection. Linguistics can reveal communication and cultural transmission. When both show island-level structure, we gain stronger evidence that geography and social networks shaped Cabo Verdean history. And where genetics and language diverge, the difference is equally interesting. It reminds us that culture can travel in ways DNA cannot. Cabo Verdean history was written in chromosomes, but it was also written in words. Cabo Verdeans display a wide range of skin pigmentation and physical appearance. That diversity is unsurprising in a population formed largely from ancestors connected with Western Africa and Southwestern Europe, regions where several pigmentation-related genetic variants historically occurred at different frequencies. But appearance is also one of the easiest ways to misunderstand ancestry. People naturally notice skin color, hair texture, facial characteristics, and other visible traits. Yet only a tiny fraction of the information inside a genome is visible from the outside. Two Cabo Verdeans can look similar while carrying different ancestry proportions. Two relatives with similar overall ancestry can also look noticeably different. Scientists have measured this relationship directly. In the 2012 population study, researchers compared measured skin pigmentation with estimated West African ancestry. They found a clear statistical association. On average, individuals with higher West African ancestry tended to have higher measured melanin levels, but the relationship was nowhere near perfect. The reported correlation was approximately 0.54. That is scientifically meaningful. It also leaves a large amount of individual variation unexplained. Skin pigmentation, therefore, contains some ancestry information statistically at the population level. It is not a reliable DNA test for an individual. Inheritance explains part of the reason. Genome-wide ancestry estimates draw information from hundreds of thousands of positions. Skin pigmentation depends much more strongly on a smaller subset of genomic regions, although many genes contribute. Every child receives a different combination of chromosomes from their parents, except identical twins. One sibling may inherit a particular combination of pigmentation-associated variants. Another may inherit a different combination. Their overall ancestry can remain similar while visible traits differ. The opposite can happen too. Two unrelated people may have similar pigmentation but considerably different genome-wide ancestry. Judging ancestry by appearance is therefore scientifically unreliable. It can also become socially harmful when visible characteristics are treated as measures of identity, authenticity, status, or human value. Cabo Verde's colonial and post-colonial history included social categories influenced by ancestry, legal status, wealth, and color. Social meanings attached to appearance can continue even after the institutions that first produced them change. Research in Cabo Verde found associations between skin pigmentation and socioeconomic indicators that were not explained simply by genetic ancestry. That distinction is crucial. Social inequality is not encoded as biological worth. If two people with similar ancestry are treated differently because one is perceived as having darker skin, the mechanism is social perception and social structure, not a difference in human capability. Genetics provides no scientific justification for such hierarchies. Another area of Cabo Verdean research demonstrates how individual genetic regions can tell a different story from overall ancestry. Researchers have examined the ACKR1 gene, historically known as DARC, which influences the Duffy blood group system. A variant commonly called Duffy null occurs at very high frequencies in many Western African populations. The variant strongly reduces susceptibility to infection by Plasmodium vivax, a parasite associated with malaria. A study of Cabo Verde found unusually high West African local ancestry around the DRC region on Santiago. At the relevant locus, West African ancestry was approximately 83.4%, compared with a genome-wide average of about 73% in the Santiago sample used in that analysis. Researchers interpreted this pattern as evidence consistent with natural selection, increasing the frequency of an African-derived protective variant after admixture. This illustrates an important principle. The genome is not only a record of migration. Environmental pressures can alter the frequencies of particular variants after populations meet. Imagine a recently admixed population containing chromosome segments from two major ancestry sources. Most segments change through inheritance, recombination, migration, and chance. But if one segment carries a variant that substantially improves resistance to an important local disease, people carrying it may become proportionally more represented in later generations. That particular region can then acquire an ancestry proportion different from the rest of the genome. Natural selection has changed one part of the mosaic. This should never be interpreted as evidence that one population is biologically superior to another. Evolutionary advantages are specific to environments and traits. A variant useful in one disease environment may make little difference elsewhere. Some adaptations even come with trade-offs. Evolution is not a ranking system. It is the continuing result of populations interacting with different environments. Cabo Verde provides an unusual opportunity to study ancestry, recent population mixture, and natural selection in a society whose demographic formation is comparatively well documented. But the evidence also gives us a warning. Skin color shows only one part of genetic history. Ancestry percentages show only one part of historical identity. Neither can reveal intelligence, personality, cultural authenticity, character, or human value. The human face is visible. The population history behind it is far more complicated. For many people, population genetics becomes personal the moment they open a commercial ancestry test. A Cabo Verdean, or someone from the Cabo Verdean diaspora might receive categories labeled Senegal, Guinea, Portugal, Spain, West Africa, Southern Europe, or something similar. Another company could produce different labels. And after a database update, the percentages might change again. That can be confusing. The first thing to understand is that commercial ancestry estimates are statistical comparisons. They are not direct measurements of nationality. Testing companies construct reference panels using customers or research participants whose ancestry is associated with particular regions. Algorithms then compare segments of a customer's genome with patterns found in those reference samples. If one segment is especially similar to Portuguese references, the company might classify it as Portuguese or Iberian. Another segment might resemble populations sampled in Senegal or neighboring parts of Western Africa. But Cabo Verde itself is an admixed population. When a company has a limited Cabo Verdean reference panel, the algorithm may divide a Cabo Verdean person's ancestry primarily among older source regions. A company with a stronger Cabo Verdean database may instead identify a Cabo Verdean genetic community directly. Neither method means that culture is encoded differently. It reflects the structure of the company's reference database. Those databases continually change. As companies acquire more samples, broad regional categories can become more specific. An estimate once labeled simply associated with Western Africa does not mean every female ancestor came from Western Africa. It means one uninterrupted mother-to-mother line points there. Likewise, a European-associated Y haplogroup does not mean that most of a man's ancestry is European. It describes one uninterrupted father-to-father pathway. This distinction becomes increasingly important deeper in the family tree. 10 generations ago, a theoretical pedigree contains more than 1,000 ancestral positions. The actual number of distinct people is usually lower because relatives appear repeatedly in family trees, a phenomenon known as pedigree collapse. And a person does not inherit identifiable autosomal DNA equally from every distant genealogical ancestor. Some ancestors contribute segments that survive clearly. Others contribute extremely small fragments. Some genealogical ancestors may eventually contribute no detectable autosomal DNA to one particular modern descendant. The genealogical relationship still existed. Genetic inheritance and family trees overlap, but they are not identical. For Cabo Verdean families, this distinction is particularly meaningful because historical documentation is uneven. A family may know it has deep Western African ancestry without possessing records identifying a specific ancestral village or community. A commercial test may reveal a strong affinity with Senegambian populations. That finding can refine the historical question. It cannot automatically reconstruct an exact ethnic identity. A person whose DNA resembles modern Mandinka reference samples cannot conclude that every relevant ancestor belonged to a Mandinka community. Historical populations moved. Modern reference samples overlapped genetically. Ethnic identities changed. Databases remain incomplete. The scientifically responsible language is one of probability and affinity. The same caution applies to European ancestry. A strong Iberian signal aligns closely with Cabo Verde's Portuguese colonial history. But it does not prove that every European ancestor was Portuguese. Atlantic maritime networks brought people from other regions as well. The most productive DNA results therefore generate new questions. Which island did the family come from? Which surnames recur in historical records? Does a direct paternal lineage connect with Iberia? Does a mitochondrial lineage show Western African affinity? Which cousins share the same segments? Can a genetic match be connected with a named historical ancestor? These questions transform ancestry testing from a percentage game into actual genealogical research. Most importantly, a DNA test cannot determine who is genuinely Cabo Verdean. Culture is not measured through genetic percentages. Someone might have deep biological ancestry in the islands but grow up with limited exposure to Kriolu or Cabo Verdean traditions. Another person might have more recent ancestry from elsewhere yet grow entirely within Cabo Verdean family and community life. Genetics can illuminate biological inheritance. Identity grows from family, language, memory, place, history, culture, and personal experience. DNA is a tool. It is not a judge. After examining settlement, autosomal ancestry, mitochondrial DNA, the Y chromosome, island differences, language, pigmentation, and commercial testing, we can return to the central question. What is the DNA of Cabo Verdean people? The strongest scientific answer is that Cabo Verdeans formed primarily from substantial ancestry related to Western Africa, especially Senegambia, and southwestern Europe, especially Iberia. But there is no single genetic percentage that defines Cabo Verde. The major 2012 study estimated mean West African autosomal ancestry of approximately 57% across the 6 islands it sampled. Yet Santiago, Fogo, Boa Vista, São Nicolau, Santo Antão, and São Vicente had different averages. Later genome-wide research confirmed that island-level variation forms part of a broader pattern of fine-scale population structure. The African component is also more informative than a vague continental category suggests. Modern analyses find particularly strong affinity with Mandinka and Wolof reference populations from Senegambia. Historical evidence shows additional connections across the Upper Guinea region and beyond. The European component is similarly structured, with Iberian reference populations providing the dominant affinity. Maternal and paternal ancestry add another dimension. The major mitochondrial study found overwhelmingly Western African direct maternal lineages. Y chromosome research revealed much greater European representation in direct paternal lines. X chromosome ancestry supports the same broad sex-biased demographic pattern. Together, the evidence describes a population that emerged during a colonial period marked by extremely unequal migration and social structures. But that is only the beginning of the Cabo Verdean story. Once communities formed in the islands, new generations began creating something that did not previously exist. Kriolu developed. Island communities expanded. Santiago and Fogo followed different demographic paths. Santo Antão, São Nicolau, Brava, Maio, Boa Vista, Sal, and São Vicente developed distinctive histories. People moved between islands. Some communities remained relatively isolated. Others became maritime centers. Drought and economic hardship encouraged migration abroad. Cabo Verdeans established major diaspora communities in Portugal, the United States, the Netherlands, France, Senegal, and elsewhere. The population continued evolving long after its earliest African-European admixture. Ancestry analysis works backward. It tries to decompose a modern genome into older sources. Human communities live forward. They receive inherited elements and turn them into something new. Cabo Verde is an exceptional example. 5 centuries ago, there was no long-established Cabo Verdean population distributed across these islands. Today there is a people with a nation, multiple Criolu varieties, musical traditions, historical memory, a global diaspora, and a detectable population genetic structure. The ancestral populations came from different regions. The society that emerged became distinct. Modern genetics can even detect differences between islands that formed after the original population mixture. Cabo Verdean genomes therefore contain more than African-related segments beside European-related segments. They also preserve centuries of specifically Cabo Verdean demographic history. Culture demonstrates the same principle even more clearly. Criolu cannot meaningfully be assigned an African percentage and a European percentage matching the genome. Neither can Morna, nor can Funana, Batuk, Cachupa, maritime traditions, family customs, or the concept of Morabiza. Culture follows different rules. It is taught, shared, modified, remembered, sometimes lost, sometimes revived, and often adopted by people whose genetic ancestry differs dramatically. That does not make DNA irrelevant. Genetics is exceptionally powerful when answering biological questions. It can illuminate migration, ancestry, founder effects, population structure, sex bias contribution, isolation, and natural selection. But genetics becomes misleading when asked to answer questions for which it was never designed. It cannot determine cultural authenticity. It cannot measure how Cabo Verdean someone is. It cannot rank one island above another. And it cannot establish that one ancestral population possessed greater human value than another. In fact, modern genetics undermines such simple divisions. Every island contains substantial individual variation. The populations labeled African and European contain enormous diversity within themselves. And every modern human genome is the product of countless ancestral pathways. Cabo Verde, therefore, teaches a wider lesson about human populations. Populations are not permanent biological boxes. They form, they migrate, they separate, they reconnect, they transform. Identities that appear ancient and fixed may actually be historically recent. Cabo Verdean identity is one such historical creation. That does not make it less real. It makes its formation unusually visible. The genome reveals where some ancestral threads began. Atlantic history explains how those threads arrived in the islands. Geography shaped how they moved between generations. And Cabo Verdean communities transformed them into a society that cannot be reduced to any one ancestral source. That is the most important conclusion hidden inside the DNA. Cabo Verdeans are not simply a mathematical combination of Africa and Europe. They are descendants of a historical process that began with multiple ancestral populations and produced something distinct in the Atlantic. The DNA of Cabo Verde does not reveal one race, one fixed percentage, or one simple origin. It reveals movement between Western Africa and Iberia. It reveals overwhelmingly Western African direct maternal lineages, alongside a much stronger European contribution in many paternal lines. It shows the effects of colonial expansion, forced migration, settlement, isolation, adaptation, and generations of movement between islands. And it reveals that Santiago, Fogo, Santo Antão, São Vicente, São Nicolau, Boa Vista, Maio, Sal, and Brava did not all follow the same demographic path. But chromosomes can only take us so far. DNA can tell us where parts of a population's ancestry came from. History helps explain how people arrived. Language and culture reveal what their descendants created. And perhaps that is the most important part of the story. Cabo Verdean identity is not a percentage inherited from the past. It is something generations of people built in the Atlantic. So if your family's DNA could recover one forgotten journey from that history, which journey would you want to discover?